first scene is not a garden, not an ocean full of blue calm, not a planet waiting patiently for life. It is a dark young world under a sky without breath, a world still glowing from its own making, where stone runs like liquid in places, where the ground shudders under bombardment, and where the moon hangs much closer than it does today, swollen and pale above a planet that has not yet learned how to be stable. Earth is young, but not gentle. Its surface is a restless skin over heat. Its atmosphere is thick with volcanic gases, steam, carbon dioxide, nitrogen, sulfur compounds, perhaps methane in some places, perhaps other gases in others, depending on which model of the ancient air scholars trust most. There is no grass, no forest, no animal movement, no birds crossing a dawn. There is only rock, fire, vapour, impact, darkness, and water trying to find a place to remain. Somewhere in this vast violence lies the beginning of the only life we know for certain ever existed. But the mystery is that the beginning does not look like life. It does not leave bones. It does not leave shells. It does not even leave a clear fossil that can be held up and called the first organism. The beginning is a blur hidden inside chemistry, and the oldest rocks are poor witnesses, burned, folded, melted, buried, lifted, and altered across almost 4 billion years. What survives in the record suggests that life may have appeared very early, perhaps earlier than the rocks can clearly tell us, but the evidence remains incomplete. That incompleteness is part of the story. The origin of life is not a clean doorway. It is a foggy shoreline where geology, chemistry, and biology begin to overlap. Before we go deeper, take a moment to like the video and subscribe. If this is the kind of history and mystery you enjoy, this story will move slowly across deep time, but it begins with speed, heat, and collision, because Earth itself begins as an accident of accumulation. Dust circles the young sun, grains become pebbles, pebbles become boulders, boulders become planetary embryos, and each collision brings more mass and more heat. The young Earth forms roughly 4.5 billion years ago, not as a finished planet but as a growing body inside a dangerous neighbourhood. Other worlds still cross its path, debris still rains through space. A giant impact, probably with a Mars-sized body, helps create the moon, and the energy of that event may have melted much of Earth's outer layers. The planet that might one day hold oceans first becomes something closer to a wound in space, glowing, spinning, releasing heat, and cooling unevenly beneath a sky of vapour. The strange thing is that the ingredients of life may already be arriving before life exists. Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, metals, and water are present in different forms, scattered through rocks, gases, and falling bodies from space. Meteorites and asteroids may have delivered some organic molecules, while Earth's own chemistry may have made others. This does not mean life came ready-made from the sky. It means the raw materials may have been widespread, perhaps common in the early solar system. Much later, samples from the asteroid Bennu would show that small bodies can preserve amino acids and nucleobases, ingredients that living cells on Earth use today. That discovery does not prove that life exists elsewhere, and it does not solve the origin of life, but it makes the early Earth feel less isolated. The planet may have been struck not only by destructive stones, but by chemical packages, ancient fragments carrying pieces of possibility. For a long time, the popular image of life's beginning has been a lightning bolt over a warm pond, a dramatic spark that suddenly turns dead chemistry into living matter. That image is memorable, but the real story is probably slower. stranger, and less theatrical. There may not have been a single place or a single moment. Instead, there may have been many chemical theatres at once volcanic shorelines, drying pools, impact-heated basins, mineral-rich vents on the seafloor, porous rocks where water and heat pass through tiny compartments, ice-covered environments where molecules became concentrated in briny pockets, and shallow landscapes where wetting and drying could push reactions forward. Scholars still debate which setting mattered most, because each offers something useful and each has problems. A deep-sea vent can provide energy and mineral surfaces, but some molecules may be too diluted in the vast ocean. A shallow pond can concentrate chemicals as water evaporates, but it must survive ultraviolet light, impacts, and chemical instability. The early Earth offers many laboratories, but none comes with a label reading "This is where life began." Water is both a gift and a complication. Life today depends on it so completely that it is difficult to imagine biology without water, yet water can also break apart fragile molecules as quickly as it helps them meet. On the young Earth, water may have condensed from the atmosphere as the planet cooled, or arrived partly through water-bearing bodies from space, or both. The exact balance remains debated. What matters for the story is that liquid water appears early enough to change the planet's character. Steam becomes rain. Rain becomes temporary pools, rivers of hot runoff, and eventually seas. The first oceans are not the calm blue surfaces familiar today. They are probably darker, more acidic in some places, richer in dissolved iron, affected by volcanic outgassing and bombardment. The sky above them is not protected by an oxygen-rich atmosphere or a modern ozone layer. Ultraviolet radiation reaches the surface with far greater force. Lightning flashes over volcanic clouds. Impacts throw heat and dust into the air. Hydrothermal systems pump hot, chemically charged fluids into the depths. If life is going to begin here, it must begin in a world that does not yet seem designed for it. That may be the most important shift in how this story should be heard. Earth is not created as a comfortable home for life. Life, if it emerges, must negotiate with the planet as it is. It must arise from local pockets of order inside a larger environment of instability. It must find energy before there is sunlight harvesting biology. It must preserve information before there are cells like ours. It must build boundaries before there are membranes refined by evolution. It must repeat chemistry with enough reliability that chance can become inheritance. Nothing about that is simple. No single explanation fully accounts for this transition, and that is why the origin of life remains one of the deepest scientific mysteries. It is not mysterious because science has no clues. It is mysterious because the clues are scattered across fields that rarely fit neatly together: geology, chemistry, planetary science, molecular biology, evolutionary theory, and the study of worlds beyond Earth. The young Earth is also changing too quickly to keep its own diary. Rocks from this earliest chapter are almost entirely gone. Plate tectonics, melting, erosion, and metamorphism have erased or rewritten much of the original crust. The planet recycles itself. The evidence we want most has passed through fire and pressure, even when ancient minerals survive. They often survive as fragments, as zircons or altered stones, carrying hints rather than full scenes. This means that the first part of life's history must be reconstructed from indirect evidence isotopes, minerals, molecular comparisons among living organisms, experimental chemistry, and models of ancient environments. It is a detective story in which the crime scene has been washed by oceans, buried under continents, and cooked inside the earth for billions of years. Yet the absence of a perfect record does not mean the past is silent. It speaks in patterns, it speaks in carbon chemistry, in the structure of living cells, in the shared genetic machinery found across all known life, in the strange universality of the genetic code, in the way organisms handle energy, and in the fact that every living thing on Earth, from bacteria in boiling vents to redwood trees, from fungi in soil to human beings listening in the dark, seems to descend from a common ancestral population. That ancestor, later called LUCA, belongs further ahead in the story. But even the idea of LUCA sends a signal backward before the familiar tree of life spreads into its great branches. Something has already crossed the threshold into biology. Something has already learned how to persist. For now, the planet is still becoming. Its oceans are forming, its rocks are cooling, its atmosphere is unstable, and its surface is being written and erased at the same time. Somewhere in its heat and water, in mineral pores or shallow basins, in reactions fed by sunlight, lightning, impacts, or geochemical gradients, chemistry begins to wander toward a new kind of behavior, not life yet, not in any clear sense, but perhaps a chemistry that repeats, a chemistry that gathers, a chemistry that makes small boundaries between inside and outside, a chemistry that starts, very slowly, to remember. The first oceans do not preserve their own first waves. They leave no shoreline that can be walked back to the morning of the world, no beach where the earliest foam hardened into stone, no tide mark that says life began nearby. Earth's oldest seas are mostly known by absence, inference, and survival through damaged fragments. The planet has been too restless, too hot inside, too determined to recycle itself. Crust forms, sinks, melts, rises, folds, erodes, and disappears. Ocean floors are especially temporary. Even today, most seafloor is young compared with the continents, constantly being created at ridges and swallowed at subduction zones. So when the mind tries to imagine the first ocean, it is imagining a world that has almost entirely vanished, a water-covered memory erased by the very planet that made it possible. Yet water changes everything. Once liquid water can remain on the surface, Earth becomes more than a cooling stone. It becomes a chemical engine with circulation, concentration, dissolution, precipitation, erosion, and exchange. Rain falls through a strange atmosphere and attacks fresh volcanic rock. Rivers, perhaps short-lived at first, carry dissolved minerals into basins. Waves grind stone into particles. Heat rises from below. Impacts punch craters into the surface and may leave behind hot basins where water reacts with shattered rock. In the deep places, seawater enters cracks, heats, transforms, and returns loaded with reduced chemicals. Above all of this, sunlight strikes a surface without the protection of a modern oxygen-rich sky. The first oceans are not passive, they are working. One of the great debates in origin of life research is where this work mattered most. Some researchers look downward, toward hydrothermal systems on the seafloor, where heat and chemistry meet in darkness. Others look toward land, toward shallow pools, volcanic ponds, tidal flats, and places where water comes and goes. The evidence remains incomplete, and no setting has solved the entire mystery. But each possible landscape reveals a different way early Earth may have pushed chemistry toward complexity. The deep sea offers steady energy, the land offers cycles, mineral surfaces offer structure, cracks and pores offer small compartments. The young planet may have provided not one birthplace, but thousands of experiments running at once, most of them failing silently. At hydrothermal vents, water and rock create powerful chemical gradients. In some settings, seawater seeps into the crust and reacts with mantle rocks, producing hydrogen through a process often linked with serpentinization. That hydrogen can feed reactions involving carbon dioxide, forming simple organic molecules under the right conditions. In alkaline hydrothermal vent models, warm mineral-rich fluids rise into more acidic ocean water, producing natural differences in chemistry across thin mineral barriers. To a modern cell, gradients across membranes are life's currency. Cells spend much of their existence maintaining imbalances, keeping one side different from another, using those differences to make energy usable. In a vent, nature may have made crude versions of such gradients before life existed. The comparison is tempting, but scholars still debate how far it can be pushed. A mineral wall is not a cell membrane. A vent chimney is not an organism. Still, the resemblance is suggestive of a world of tiny compartments, flowing chemicals, and energy waiting to be captured. These mineral compartments may be one of the lesser-known keys to the story. Life depends on boundaries. Without some division between inside and outside, useful chemistry drifts away. A cell is, among other things, an agreement that certain molecules belong together long enough to do work. Before cells, porous rocks could have created temporary neighborhoods for molecules. Iron sulfur minerals, clays, carbonates, and other surfaces might have attracted, aligned, or protected certain compounds. A surface can bring molecules close enough to react. A pore can trap them. A crack can allow fresh fluid to enter while keeping some products from vanishing immediately into the open sea. This does not mean rocks were alive. It means that minerals may have helped chemistry behave less randomly. The shallow water models offer a different kind of order. Imagine a volcanic landscape under a harsh sky, with pools fed by rain, steam, runoff, and geothermal fluids. Some pools dry in heat, concentrating whatever molecules they contain. Later, rain returns. Wetting and drying cycles can help link small molecules into longer chains, while full immersion can separate, transport, or reshape them. The rhythm matters. Modern biology is full of cycles day and night, wet and dry, growth and rest. Energy gained and energy spent. On the early Earth, before any organism could regulate itself, the environment may have imposed cycles from outside. A pool that dries at its edges is not a gentle cradle, but it can do one important thing the ocean struggles to do: it can concentrate. That problem of concentration haunts almost every origin scenario. The ocean is enormous. Dilution is unforgiving. Molecules that might react in a laboratory can become hopelessly scattered in open water, shallow basins, evaporating pools, mineral films, ice pockets, and porous vents all offer possible ways around that problem. Even ice, which seems hostile to life, can create tiny briny channels where dissolved compounds become crowded together. Dormancy, or chemical weighting, may also matter more than once seemed obvious. Life today uses inactive states to survive extremes, and while it would be wrong to project modern biological dormancy back onto lifeless chemistry too directly, the broader principle is useful persistence may have required not constant activity, but the ability of fragile systems to pause, endure, and resume when conditions improved. Impacts complicate the scene further. For older narratives, bombardment often appears mainly as destruction, a reign of violence preventing early life from gaining a foothold. But impacts are not only erasers, they also fracture crust, melt rock, create hydrothermal systems, deliver materials, excavate minerals, and generate environments that can last for thousands or even millions of years. A large impact can sterilize a region. It can also create a hot chemical reactor. The early Earth may have been repeatedly damaged and repeatedly made chemically interesting. That does not make life inevitable. It only means that catastrophe and possibility were not always separate forces. On this young planet, they often arrive together. The tragedy for anyone trying to read this world is that nearly all direct evidence from this chapter is gone. The rocks that might have held the clearest signs of prebiotic chemistry have mostly been transformed beyond recognition or destroyed entirely. Ancient zircons, some older than 4 billion years, hint that liquid water may have existed surprisingly early, but a zircon is not an ocean, and it is certainly not a cell. It is a mineral survivor, a hard little witness that carries chemical clues from a vanished crust. Other rocks from later periods preserve possible signs of early biological activity, but those belong to a time after the deepest origin mystery has already passed behind the curtain. The very first steps, if they occurred before the oldest clear biological evidence, are largely inaccessible. This is why the first oceans feel almost mythic, even when described scientifically. They are not myth because they are false. They are myth-like because they stand at the edge of evidence, where the known world thins into reconstruction. Scientists can test reactions, compare living cells, study meteorites, model atmospheres, analyze isotopes, and search ancient rocks. But no one can replay the first successful transition from geochemistry to biology. What survives in the record suggests that Earth became habitable early, and that life may have followed with surprising speed, but the exact path remains uncertain. The first oceans remember nothing in the way a fossil remembers a shell or a footprint remembers a passing animal. They remember through chemistry, through altered stone, through the deep habits of life that still exist. And so the story stays suspended over several landscapes at once, black vents breathing into iron-rich seas, warm pools shrinking under volcanic skies, mineral pores holding molecules in darkness, impact basins steaming after violence, icy pockets concentrating chemistry in cold silence. None can yet claim to be the birthplace. All may have mattered. Across them, the same problem repeats in different forms: how to make fragile molecules last, how to bring them together, how to let energy flow through them, how to build a boundary, how to create a pattern that can survive long enough to be copied. The first oceans do not remember the answer clearly, but they give the setting in which the answer must have begun to form. Chemistry does not become biology simply by becoming complicated. A tarry mixture of carbon-rich molecules can be complex and still be lifeless. A pool can contain amino acids, sugars, fatty acids, phosphates, metals, and countless reaction products without producing anything that eats, grows, copies itself, or evolves. The hard step is not only making ingredients, it is making organization. Somewhere in the lost chemistry of the young Earth, molecules had to stop behaving like scattered debris and begin taking part in systems, where one reaction helped another, where energy was not merely released as waste heat, but captured and redirected, where certain patterns lasted longer than others, and where the difference between accident and inheritance slowly began to matter. This is the quiet turning point hidden beneath all later history, long before eyes, shells, bones, forests, or even true cells. There must have been a time when chemistry learned to hold itself together. The ingredients themselves are not the greatest surprise anymore. That does not mean they are simple to explain, only that science has found many plausible paths to them. Organic molecules can form in space, in meteorites, in hydrothermal systems, in atmospheric reactions, in ice, on mineral surfaces, and in laboratory simulations designed to mimic pieces of the early Earth. The presence of amino acids and nucleobases in primitive asteroid material reminds us that the solar system was chemically rich before biology existed. But an amino acid is not a protein, and a nucleobase is not a genome. It is easy to confuse the alphabet with the book. The early Earth may have received letters from space and made others on its own, but life required grammar, copying, boundaries, and a way to keep going. That need for activated chemistry is one of the lesser known but crucial parts of the story. Many molecules do not simply link themselves into useful chains in water. Water can help them move and meet, but it can also cut them apart. To build longer structures, chemistry often needs energy stored in reactive bonds. Modern cells use elaborate machinery to do this with astonishing precision. But before enzymes, before ribosomes, before the elegant systems inside even the simplest living cells, there must have been rougher chemical shortcuts. Thioesters are one possible clue. In modern metabolism, thioester chemistry still appears in important energy handling processes, almost like an ancient accent preserved inside the language of cells. Recent work suggests that amino acids activated by thioesters can attach selectively to RNA in water without enzymes, offering a possible bridge between prebiotic chemistry and the later world of protein synthesis. This does not solve the origin of life, and scholars still debate how such chemistry would have operated in real, early Earth environments, but it shows something important. Water does not always have to be the enemy of organization. Under the right conditions, even in water, chemistry can become selective. Selectivity matters because life is not just chemistry that happens. It is chemistry that chooses, though not consciously, of course. Some molecules fit together better than others. Some surfaces hold certain compounds more strongly. Some reaction networks regenerate their own starting materials. Some fragile structures fall apart instantly, while others persist long enough to encounter new conditions. Before natural selection in the familiar biological sense, there may have been a kind of chemical selection. Molecules that formed easily, lasted longer, accumulated in favourable places, or helped produce more of themselves would become more common. No intention is needed, no plan is hidden behind it. A tide pool does not know what it is doing. A mineral surface has no purpose, but repeated conditions can make repeated outcomes, and repeated outcomes are the first faint shape of order. The early Earth may have been full of such partial orders. Fatty molecules can naturally form droplets, vesicles, or membrane-like structures under some conditions, creating small enclosed spaces. These boundaries are primitive, leaky, unstable, and far from living cell membranes. But they introduce a radical idea: an inside. Once there is an inside, even a temporary one, chemistry can be protected from dilution. Molecules can accumulate together. Reactions can influence one another. A vesicle that traps useful molecules may behave differently from one that traps only useless sludge. If it grows by absorbing more fatty material and divides when disturbed by waves or heat, it begins to resemble a crude physical model of reproduction, though not true life yet. The evidence remains incomplete, and no one should imagine a perfect little cell suddenly forming from a puddle. But boundaries could have given chemistry a stage small enough for organization to matter. Minerals may have acted as another kind of stage. Clays can absorb organic molecules and arrange them along surfaces. Iron-sulfur minerals can participate in electron transfers. Metal ions can stabilize certain structures or catalyze reactions. In hydrothermal settings, natural compartments can keep fluids separated while allowing energy to flow. On sunlit land, ultraviolet radiation can destroy delicate compounds, but it can also drive useful reactions that would not happen easily in darkness. In drying pools, molecules can become concentrated into films. In ice, briny channels can crowd chemistry into narrow spaces. No single environment supplies every answer, and that is precisely why the origin of life may not have been a single-scene event. It may have been a network of places, with molecules moving between them, altered by one setting and tested in another. Energy is the thread that ties these settings together. Life is not merely matter arranged in a special way. It is matter through which energy flows. A dead cell and a living cell can contain nearly the same atoms, but one is maintaining gradients, repairing damage, building molecules, and resisting equilibrium, while the other is surrendering to chemistry's downhill slide. For life to begin, some pre-living system had to tap energy reliably. It might have used redox gradients near vents, sunlight-driven reactions at the surface, chemical energy from activated molecules, or thermal cycles that pushed compounds through phases of assembly and breakdown. What survives in modern biology suggests that energy handling is not a late decoration. It lies near the heart of life. Even LUCA, when it appears later in the reconstruction, seems to have been metabolically active, not a bare genetic ghost floating alone. That hints that metabolism, or something like it, may have been deeply ancient. There is a famous divide in origin of life thinking between metabolism first and genetics first. Though the real history may not respect that neat division, metabolism first models imagine networks of reactions becoming self-sustaining before genetic molecules take command. Genetics first models emphasize molecules capable of storing and copying information. Both sides capture something essential. A system without energy flow cannot persist, but a system without information cannot evolve in the rich biological sense. The earliest life may have required both, emerging not as a finished machine but as a messy partnership between reaction networks, boundary-forming molecules, and information-bearing polymers. It is possible that several half-systems formed many times and failed many times before one combination became durable enough to leave descendants. This is where the story becomes difficult to picture, because the familiar categories break down. A molecule is not alive, a vesicle is not alive, a reaction cycle is not alive. A strand that helps copy itself is closer, but still not necessarily life. The boundary may have been crossed gradually, and our modern definitions may be too sharp for the ancient transition. Biology today is full of clean labels because billions of years of evolution have refined the machinery. At the beginning, the machinery was not refined. It may have been noisy, leaky, inefficient, and chemically ambiguous. The first systems capable of evolution may have looked less like tiny versions of modern cells and more like unstable chemical collectives, surviving only in favorable pockets, constantly threatened by dilution, heat, ultraviolet light, and their own errors. Still, even unstable collectives can change the future if they begin to reproduce with variation. Once copying exists, however crude, time becomes powerful. Errors are no longer just failures. Some become differences that selection can test. Compartments that grow more reliably become more common. Molecules that help build or copy useful structures persist. Reaction networks that waste less energy outlast others. The planet does not need to intend anything. It only needs countless trials and enough time for rare successes to accumulate, And on the early Earth, time is already opening in immense quantities, not years or centuries, but millions upon millions of years under volcanic skies. So chemistry learns to organize not in one miracle, but through many small advantages, a bond that stores energy, a surface that holds molecules close, a cycle that repeats, a droplet that traps the right mixture, a chain that influences its own formation, a boundary that lets an inside remain different from an outside. Each step alone is not life. Together, under conditions we're still trying to reconstruct, they begin to point toward the threshold. Somewhere beyond that threshold, the story will no longer be only about molecules, it will be about ancestry. The threshold between organized chemistry and true ancestry narrows around one strange molecule, a molecule that still lives inside every cell today, but may also carry the echo of a much older world. RNA is not as famous as DNA in popular imagination, and it does not have the same sculptural elegance of the double helix. It is usually presented as a messenger, a temporary copy, a working note passed from DNA to the machinery that builds proteins. But that modern role may hide an ancient importance. RNA can store information in the sequence of its bases, and under some conditions, RNA can also fold into shapes that perform chemical work. It is both message and machine, or at least it can be. That double identity makes it one of the most important suspects in the mystery of how life began. The idea called the RNA world does not claim that modern RNA simply appeared fully formed on the young Earth. That would be too easy, and almost certainly wrong. Instead, it suggests that before DNA became the long-term archive of life, and before proteins became the great workers of the cell, RNA-like molecules may have played both roles in rougher form. They may have carried information, helped copy themselves or related molecules, and catalyzed reactions inside primitive chemical systems. The evidence remains incomplete, because no fossil strand of RNA from 4 billion years ago can survive. RNA is fragile, it breaks, it reacts. It does not wait patiently in stone for a future scientist. So the RNA world is reconstructed not from direct remains, but from chemistry, laboratory experiments, and living cells themselves, which preserve clues in their deepest machinery. One of the strongest clues is hidden in the ribosome, the molecular machine that builds proteins in every known living cell. The ribosome is enormous and complex, but at its catalytic heart, the part that actually helps form the bond between amino acids. RNA does the central work, proteins surround and support the structure, but the core chemistry is performed by ribosomal RNA. This is a surprising detail, almost like opening a modern factory and finding that its oldest, most essential tool is made from a material once thought to be only a set of instructions. In that sense, every cell carries a relic of an RNA-centered past. It does not prove the entire RNA world exactly as imagined, but it gives the hypothesis a powerful reason to exist. RNA is built from nucleotides, and each nucleotide contains a sugar, a phosphate, and a base. In modern biology, assembling these pieces requires sophisticated enzymes and carefully controlled conditions. On the early Earth, none of that machinery existed. This is one of the great problems. It is not enough to say RNA could store information and catalyze reactions. One must also explain how RNA-like molecules could form in the first place, how their building blocks could accumulate, how the right chemical forms could be favored, and how strands could grow long enough to matter without being destroyed. Scholars still debate these steps intensely. Some propose that RNA was preceded by simpler genetic polymers, molecules not identical to RNA, but capable of similar information storage. Others argue that networks of chemistry could have gradually produced RNA components, under changing environmental conditions. The true pathway may have been messier than the name RNA world suggests. The word world can also mislead. It does not necessarily mean the entire planet was covered in RNA life. It means a stage, a regime, a period in which RNA-like chemistry played a central evolutionary role. It may have existed in certain environments only, in pools or pores or mineral surfaces where ingredients gathered and cycles repeated. A strand of RNA alone in the open ocean would be nearly helpless, diluted, exposed, and chemically vulnerable. But inside a protected microenvironment, perhaps near mineral surfaces or within fatty vesicles, RNA-like molecules could have gained time. Time is crucial. A molecule that lasts only moments is an event. A molecule that persists, folds, reacts, and influences its own copying begins to become history. The copying problem is the heart of the matter. For Darwinian evolution to begin in a meaningful way, information must be passed on with variation. Copying must be accurate enough to preserve useful patterns, but imperfect enough to create differences. If copying is too sloppy, information dissolves into noise. If it is perfectly fixed, adaptation stalls. Modern cells solve this with complex enzymes and repair systems, but the early Earth had no such luxury. Experiments have shown that ribozymes, catalytic RNA molecules, can perform many kinds of reactions, including steps related to copying and ligation, but creating a fully self-sustaining RNA replication system under plausible prebiotic conditions remains difficult. No single explanation fully accounts for this. The RNA world is compelling not because it answers every question, but because it places information and catalysis in the same molecule, reducing a problem that otherwise seems divided between genetics and metabolism. There is also the issue of selection among molecules. Not every RNA sequence folds into a useful shape. Many do nothing special. Some fold badly. Some might catalyze a reaction weakly. A rare few may help create conditions that allow their own sequence, or related sequences, to become more common. In a modern laboratory, scientists can select functional RNA molecules from vast pools of random sequences, showing that RNA has a remarkable chemical flexibility. But the early Earth was not a laboratory bench with purified reagents and patient technicians. It was dirty, variable, mineral-rich, salty, acidic here, alkaline there, hot in one place, freezing in another. For the RNA world to matter, useful RNA-like chemistry had to survive in that messy planet, not in an idealized flask. Yet messiness can sometimes help. Cycles of drying and wetting may help build longer chains. Freezing can concentrate molecules in briny veins. Mineral surfaces can bring compounds together. Ultraviolet light, while destructive, may also drive certain reactions and eliminate unstable mixtures. Heat can both accelerate chemistry and ruin it. The same force can help in one setting and destroy in another. Origin of life research often feels like this because early Earth did not offer one clean condition called habitable. It offered gradients, transitions, edges, and cycles. Life may have begun not at a stable center but at boundaries between wet and dry, hot and cool, rock and water, light and shadow, inside and outside. Amino acids enter this story as partners waiting in the wings. RNA alone may store information and catalyze reactions, but proteins eventually become life's most versatile machines. The path from an RNA-centered system to a protein-building system is one of the deepest puzzles in biology. Recent work on activated amino acids and RNA offers a glimpse of how the bridge might have begun under certain conditions. Amino acids linked to energy-rich groups can attach selectively to RNA in water without enzymes. This is not yet translation, not the genetic code, not a ribosome, but it is a hint of intimacy between RNA and amino acids before modern biology. It suggests that the later partnership between nucleic acids and proteins may have roots in chemistry older than cells. At some point, RNA-like molecules may have become surrounded by boundary-forming compounds, caught inside protocellular compartments where their effects could be selected more clearly. If a strand helped build or stabilize the compartment that contained it, that compartment might persist. If another helped bring amino acids into useful arrangements, it might gain a chemical advantage. If copying improved, even slightly, selection would have more material to work with, the early RNA world. If it existed, was not a clean empire of elegant molecules. It was likely fragile, error-prone, and temporary in many places. Most attempts would have failed. Most lineages, if they can even be called that, would have vanished without descendants. What matters is that at least one chain of chemical inheritance did not vanish. The deeper one looks, the more the boundary between molecule and organism becomes difficult to draw. RNA may have been a bridge across that boundary, not because it was alive by itself, but because it made life's two hardest demands begin to meet memory and action. It could carry a pattern, and it could influence chemistry. In the long darkness before DNA genomes and protein enzymes, that may have been enough to open a path. The first true cells are not here yet. The last universal common ancestor is still ahead. But the story has changed. Chemistry is no longer only reacting, it is beginning to carry instructions, however crude, and once matter begins to remember its own arrangements, the planet enters a different kind of time. The first cell is almost impossible to picture because every living cell known today is already the product of an immense inheritance. Even a bacterium, small enough to vanish under a fingernail, is not simple in the way people sometimes imagine. Inside it are membranes, molecular readers, repair systems, energy pathways, and chemical routines refined across billions of years. To look at a modern microbe and call it primitive is a mistake. It is ancient in lineage, but not primitive in design. The first cells must have been rougher, leakier, less efficient, and more vulnerable than anything alive today. They were not tiny perfect machines. They were experiments with skins. A cell begins with a boundary, but a boundary alone is not enough. Oil droplets have boundaries. Soap bubbles have boundaries. Mineral pores have boundaries. What matters is that a living boundary helps maintain a difference between an inside and an outside, and that difference becomes useful. Inside, molecules can remain together long enough to react. Outside, the wider environment supplies raw material and energy. Modern cell membranes are built mainly from phospholipids, arranged in layers that are flexible, selective, and deeply integrated with proteins. But early membranes may have been made from simpler fatty acids or related amphiphilic molecules, compounds with one end that likes water and another that avoids it. Under certain conditions, these molecules naturally gather into vesicles, little spheres with watery interiors. They are not alive, but they do something life cannot do without they create a private space. In the young Earth's restless environment, such vesicles may have formed, broken, reformed, merged, and divided countless times. Waves could tear them apart, heat could reshape them, minerals could stabilize them, drying and wetting cycles could concentrate their contents. Some vesicles would trap nothing useful, others might enclose RNA-like strands, activated amino acids, metal ions, or small reaction networks. Most would fail in silence, dissolving back into the chemistry around them. But a few combinations may have lasted longer. If a membrane happened to contain molecules that helped it grow, stabilize, or divide, it gained a faint advantage. If its contents could be copied imperfectly and passed into daughter compartments, something closer to biological inheritance began to appear. This is one of the most delicate transitions in the entire story. The evidence remains incomplete, and there may never be a fossil of the first protocell. Still, experimental work shows why the idea is plausible. Fatty acid vesicles can grow by absorbing more membrane material. They can divide under physical forces. They can allow some small molecules to pass through while keeping larger molecules trapped. In a world before sophisticated membrane proteins, leakiness may not have been a flaw. It may have been necessary. A perfectly sealed protocell would starve unless it already possessed transport machinery. A leaky protocell could let nutrients enter while still keeping larger informational molecules inside. Metabolism enters the picture not as a finished network, but as a set of recurring chemical habits. Modern metabolism is a cathedral of pathways, with enzymes guiding reactions in exquisite sequence. Early metabolism may have been more like footpaths worn into a landscape by repeated use. Some reactions could have been driven by minerals, heat, ultraviolet light, redox gradients, or activated chemical groups, carbon dioxide and hydrogen, common in some early environments, may have fed simple carbon-fixing reactions. Sulfur and iron chemistry may have helped move electrons. Acetyl-like compounds, thioesters, and other energy-rich molecules may have acted as chemical currencies before the later dominance of ATP. No single explanation fully accounts for how metabolism began, but life could not wait until all its machinery was elegant. It had to start with rough energy flow. Some of the deepest features of modern cells still point toward very old chemistry. Cells today use proton gradients across membranes to make ATP, their central energy molecule. They rely on metal cofactors, sulfur chemistry, phosphate chemistry, and carbon reactions that may echo ancient geochemical settings. This does not mean that modern pathways are unchanged fossils from the origin of life. Evolution rewrites constantly. But what survives in biochemistry suggests that early life learned to exploit imbalances in the world before it learned to build all imbalances for itself. A vent, a mineral pore, a sunlit pool, or a chemical gradient could supply what a protocell did not yet know how to make. Replication had to become linked with these compartments. A naked self-copying molecule, if such a thing existed, might spread its information into the environment without building a durable lineage. A protocell without heritable information might grow and split, but its descendants would not improve through selection in any meaningful way. The crucial step is coupling the information inside affects the survival and reproduction of the container, and the container protects and transmits the information. Once that coupling exists, selection can act on packages rather than isolated reactions. Some protocells grow faster, some copy better, some survive heat, salt, acidity, or drying. Some fail. The world begins to sort them. Error would have been everywhere. Modern organisms use proofreading enzymes and repair systems because copying molecules is difficult. Early cells, or pre-cells, likely made mistakes constantly. Too many mistakes would erase useful information, but some mistakes would create variation. This balance between stability and change is one of life's oldest tensions. A protocell that copies too poorly loses itself. A protocell that never varies cannot adapt. Somewhere in the early biosphere, systems had to find a tolerable middle ground, not a perfect one, just good enough to persist. At this stage, life may still not have looked like a single clean lineage. there may have been many protocellular populations, exchanging molecules, merging, dividing, borrowing innovations, and losing them. The early tree of life may have looked less like a tree and more like foam, with bubbles joining and breaking apart. Horizontal gene transfer, so common among microbes today, may preserve a distant memory of that older world, where genetic identity was porous and communal. Scholars still debate how sharp the first lineages were. The first true cells probably emerged when three great needs became locked together: a boundary that could persist, a metabolism that could harvest energy, and an information system that could be copied. None of these had to be modern. The membrane could be leaky. The metabolism could be mineral-assisted and inefficient. The genome could be RNA-like or transitional. The proteins, if present, could be short and crude. What mattered was not perfection, but continuity, a system that can continue. Reproduce, vary, and leave descendants has crossed into evolutionary history. Even then, these earlier cells would have been almost invisible to geology. They would leave no skeleton, no shell, no obvious body. If they lived in warm pores, vents, sediments, or shallow waters, their remains would be chemical whispers at best. Most would vanish completely, yet from such small, fragile compartments, every later forest, coral reef, fungus, insect, whale, flower, and human mind would eventually descend. The first cells did not conquer the planet with size or strength. They survived by making an inside, keeping a pattern, and turning the restless energy of Earth into persistence. Somewhere after that, not necessarily long after and not necessarily in a single place, the lineage leading to all surviving life begins to narrow toward a common ancestry. But that ancestor is not the first cell. It is what remains visible from the far side of extinction, the last shared root of living descendants after earlier experiments disappeared. Before that root, there may have been many beginnings, many failures, and many forms of almost life. The first cells appear not as a triumphant arrival, but as tiny fragile continuities in a violent world, thin-walled pockets of chemistry that finally learn how not to vanish. Once life exists, the planet does not suddenly become kinder. The first cells do not inherit a blue world of easy sunlight and stable seasons. They inherit a restless earth, still shaped by heat from below and violence from above. A world where oceans may be dark with dissolved metals, where the atmosphere offers no oxygen to breathe, where ultraviolet light strikes the surface with little protection, and where impacts still arrive often enough to remind every fragile system that survival is not guaranteed. If these early cells are alive, they are alive in a place that seems almost hostile to the later imagination. They do not swim beneath coral reefs. They do not drift among seaweed. They belong to a microbial earth chemical, mineral, smoky, metallic, and almost entirely invisible. The most important thing about this early life is that it is not yet the world of animals, plants, or fungi. It is not even the world of complex cells. It is a world of prokaryote-like organisms, small cells without nuclei, living from chemistry in ways that can feel alien but are still echoed by microbes today. Some may draw energy from hydrogen, sulfur, iron, methane, or carbon compounds. Some may live in sediments, mineral pores, hydrothermal systems, shallow mats, or warm coastal basins. Some may survive where sunlight reaches, while others may never need sunlight at all. The evidence remains incomplete, but what survives in modern microbial diversity suggests that early life was already experimenting with several ways of making a living long before the planet had anything like a familiar biosphere. This is where LUCA begins to haunt the story. LUCA is the last universal common ancestor, the shared ancestral population from which all known living things today appear to descend. It is tempting to imagine LUCA as the first organism, a lone pioneer cell floating in the ancient sea. But that picture is misleading. LUCA is not the first life. LUCA is the last common root that still has surviving descendants. Earlier lineages may have lived and vanished completely, leaving no branch that reaches the present. Recent reconstructions suggest that LUCA may have lived astonishingly early, perhaps around 4.2 billion years ago, and may have been more complex than older simplified images implied. It may have had a fairly large set of genes, perhaps thousands of protein families in some estimates, and a metabolism resembling that of anaerobic prokaryotes, possibly linked to acetogen-like ways of using carbon dioxide and hydrogen. Scholars still debate the timing and details, because molecular clocks are difficult to calibrate across such depths of time, but the idea is powerful. By the time the surviving tree of life has a last common ancestor, life may already belong to an ecosystem, not to a single lonely cell. That possibility changes the mood of early Earth. Instead of one miraculous speck of life surrounded by dead ocean, there may have been communities of microbes, exchanging molecules, competing, cooperating, failing, and adapting in local environments. LUCA may have lived among other organisms whose descendants did not survive. The ancient biosphere, if it can be called that, may have been more like a dim microbial archipelago than a single origin point scattered habitats connected by water, rock, chemistry, and gene flow. Some lineages might have been erased by later events. Some might have merged through horizontal gene transfer. Some might have contributed pieces to the lineages that survived, then disappeared as independent forms. The early tree of life may have grown out of a tangled thicket. Survival in such a world requires more than growth. It requires endurance. Heat can denature molecules. Ultraviolet light can damage genetic material. Salts, acidity, metals, and changing chemical conditions can break fragile systems. Impacts can boil local waters, bury habitats, or transform landscapes. Volcanic activity can poison one setting while feeding another. Yet microbes have a talent that later large organisms often lack. They can live small, fast, and close to chemistry. They can occupy cracks, films, sediments, and gradients. They can endure by slowing down. The idea that dormancy or inactive states may be deeply ancient is especially suggestive. Modern microbes can pause their metabolism, form resistant states, or wait through conditions that would destroy more active life. It would be too strong to claim that the earlier cells possessed modern dormancy systems exactly as they exist today. But the broader strategy may be old. Sometimes persistence means not constant struggle, but the ability to become quiet until the world becomes favourable again. This quiet endurance may have mattered during the tail end of heavy bombardment. The old picture of early impacts as a single sterilizing storm has softened in many scientific discussions, though large impacts remain profoundly destructive. The planet could be wounded again and again without necessarily erasing every refuge. Deep crustal habitats, hydrothermal systems, buried sediments, and protected pores might have shielded microbes from the worst surface conditions. If life had already begun before some of these events, it may have survived not by standing in the open, but by retreating into the planet's hidden architecture. Rock itself could become shelter. Darkness could become protection. Heat, depending on degree and context, could become either enemy or fuel. In this microbial age, the atmosphere is not yet an oxygenated ocean of air. Oxygen, The gas later animals will depend on is rare and chemically dangerous. Many early organisms probably live without it and may even be harmed by it. Their world is anaerobic. Energy comes from redox reactions, from moving electrons between substances that the early Earth supplies in abundance. This is one reason microbial metabolisms can feel like geology learning to breathe. A microbe that uses hydrogen and carbon dioxide is participating in the chemistry of vents and rocks. A microbe that transforms sulfur or iron is altering minerals as it lives. Life is not merely sitting on Earth. From early on, it is beginning to change Earth's chemistry, subtly at first, then more profoundly than any animal ever will. One lesser-known part of this story is how small life can have planetary consequences long before it becomes visible. Microbes do not need bones or bodies to transform a world. They can shift isotopes, precipitate minerals, alter ocean chemistry, change atmospheric gases, and create layered mats in shallow waters. A microbial mat is not dramatic like a forest, but it is a living landscape, a thin, organized community where different microbes occupy different layers according to light, chemistry, and energy. Some layers may use sunlight, others may feed on byproducts from above, others may transform sulfur, iron, or carbon compounds. Even before oxygenic photosynthesis changes the atmosphere, microbial communities can create structured ecosystems in miniature. And still, nearly all of this passes without leaving clear fossils. A cell without a hard part is easily lost. A mat may leave textures or chemical traces, but those traces can be altered by heat and pressure. Carbon isotopes may suggest biological activity, but non-biological processes can sometimes mimic lifelike signatures. Ancient rocks may contain structures that look biological and later turn out to be ambiguous. This is why the early record demands caution. It is not that scientists lack imagination, it is that ancient stone can deceive. To identify life in rocks older than 3.5 billion years, researchers must ask whether shape, chemistry, context, and geology all point in the same direction. Even then, uncertainty often remains. Yet by the time clearer evidence begins to appear later, life is already established enough that its origin must lie deeper still. The first microbial world has done the most difficult thing any biosphere can do. It has persisted. It has crossed from fragile chemical beginnings into continuity across geological time. It has survived a planet without breathable air, without an ozone shield, without stable continents as we know them, without any large organism to shelter it or spread it. In darkness, in mineral pores, in sediments, in shallow mats, perhaps in hot waters and quiet buried places, life learns the art that will define it for 4 billion years. It adapts not by conquering the planet all at once, but by finding narrow opportunities and refusing to disappear. For a long time, life on Earth is almost entirely a private arrangement between microbes and chemistry. Cells live by drawing energy from reactions the planet already offers, hydrogen meeting carbon dioxide, sulfur changing form, iron accepting or giving up electrons, methane moving through sediments and water. These metabolisms are ingenious, but they are also bound to local supplies. A vent can feed life. A mineral-rich sediment can feed life. A chemical gradient across rock and water can feed life. But the greatest energy source in the solar system is shining overhead every day, pouring down on the young planet with a force no vent can match. The strange thing is that life does not seem to use it fully at first. Sunlight is present from the beginning, but it takes evolution to turn sunlight into a living economy. Photosynthesis is often imagined as green leaves under a blue sky, but that is a very late image. The first photosynthesizers are not plants. They are microbes, and many of them do not produce oxygen. Long before forests, long before seaweed, long before any leaf opens to morning light, bacterial cells discover ways to use pigments to capture solar energy and push electrons through chemical systems. This early photosynthesis may have used substances such as hydrogen sulfide, ferrous iron, or other electron donors instead of water. Its byproducts were not the oxygen-rich breath of later worlds. Some photosynthetic bacteria still live this way today in lakes, sediments, hot springs, and other layered environments where light and chemistry overlap. They are not living fossils in a simple sense, because they too have evolved, but their metabolisms help reveal how sunlight could enter biology before the atmosphere was transformed. The invention is profound because it loosens life from the strict geography of vents and chemical springs. A microbe that can use sunlight can live wherever light, water, and the right raw materials meet. It can spread through shallow seas, wet surfaces, coastal flats, and microbial mats. It can build communities in layers, with sunlit cells above and darker chemical metabolisms below. A microbial mat becomes a tiny city without streets, pigments at the surface catch light. Waste products from one layer feed another. Chemical conditions shift millimeter by millimeter. Cells divide, die, stick, trap sediment, and create textures that may survive as stromatolites. These layered structures are among the most evocative traces of early life, because they show that microbes were not merely floating specks. They were shaping landscapes, one film at a time. Still, the great transformation has not yet happened. For that, life must learn a more difficult trick using water as an electron source. Water is abundant, but it is chemically stubborn. Splitting it requires powerful molecular machinery. When certain microbes, ancestors or relatives of cyanobacteria, evolve oxygenic photosynthesis, they gain access to an almost limitless supply of electrons, because water is everywhere. The waste product of this process is oxygen. At first, the waste does not make the sky breathable. It does not immediately turn the planet blue in the modern sense. Oxygen released by microbes meets a world full of substances ready to consume it. Dissolved iron in the oceans reacts with oxygen and precipitates as iron minerals. Reduced volcanic gases absorb it. Rocks and seawater act like vast chemical sinks. For a long time, oxygen may be produced locally and destroyed locally, leaving only faint signals. This delay is one of the most important details in the story. The invention of oxygenic photosynthesis and the oxygenation of the atmosphere are not the same event. Scholars still debate exactly when oxygen-producing photosynthesis began, how widespread it was before the Great Oxidation event, and why atmospheric oxygen rose when it did. Some evidence suggests oxygenic photosynthesis may have evolved well before oxygen accumulated in the air. That means Earth may have hosted oxygen-producing microbes for hundreds of millions of years, while the atmosphere remained mostly oxygen-poor. The planet had to be chemically prepared, or exhausted, before oxygen could escape its sinks and begin to accumulate. In the oceans, one of the most dramatic witnesses to this slow preparation is the banded iron formation. These rocks, with alternating layers rich in iron minerals and silica, preserve a memory of ancient seas where dissolved iron was abundant and oxygen began to change its fate. The bands are not simple diary pages, and their formation involves complex ocean chemistry that researchers continue to study. But they remain among the most visible signs that microbial life and planetary chemistry had become entangled. In a world without animals, without plants, without any creature large enough to notice the sky. Microscopic cells are helping paint minerals across the seafloor. Life is becoming a geological force. This is also a story of colour. The ancient oceans may have held greenish, grey, or iron-dark tones in some regions, not the clear, familiar blue of modern imagination. Microbial mats may have been purple, brown, orange, black, or green depending on pigments and chemistry. The evidence for global colours is uncertain, but locally the early biosphere was likely far more visually strange than the word microbial suggests. Dark volcanic rock, iron-rich water, steaming pools, crusted minerals, and thin living films catching sunlight in colours that do not belong to grass. Oxygenic photosynthesis also changes the relationship between life and time. Before it, many metabolisms depend on supplies that can be limited by geology. After it, life taps the daily flood of solar energy through the most abundant liquid on the surface. That does not make evolution fast in a human sense. The transformation still unfolds across immense spans, but it creates a new scale of possibility. More energy can support more biomass. More biomass can alter more chemistry. More oxygen production can eventually overwhelm older sinks. A feedback begins to form between microbes and planet. These cells adapt to Earth, and Earth adapts, unwillingly, to the cells. There is a quiet irony here. Oxygen will later become the gas of animal life, the invisible partner of movement, nerves, muscles, and large bodies. But in the beginning, oxygen is waste. It is a byproduct, a dangerous reactive substance released by microbes simply because their way of harvesting sunlight produces it. To organisms built for an oxygen-free world, it can be poison. It reacts with delicate molecules, damages cells, and changes the chemistry of environments that earlier life had mastered. The very success of cyanobacteria-like organisms prepares a crisis for much of the biosphere that came before them. Life is not always gentle to life. Sometimes evolution's greatest invention is also an ecological disaster for those unable to follow it. Yet the planet does not change overnight. Oxygen leaks into microenvironments first. A mat may have oxygen in its sunlit upper layer, while the sediment below remains anoxic. Shallow waters may experience temporary oxygen oases long before the atmosphere as a whole changes. Microbes living near these boundaries face new dangers and new opportunities. Some evolve defenses against oxidative stress. Some learn to use oxygen's reactivity for energy. Others retreat into oxygen-free refuges where their descendants still live today. The biosphere becomes more layered, more divided, more chemically complex. By the time oxygen begins to matter on a planetary scale, life has already rewritten the rules of habitability. The young Earth began as a place where chemistry allowed life to appear. Now life is becoming one of the forces that determines what Earth is. The sky, the ocean, the minerals, and the future of evolution are being altered by cells too small to see. Photosynthesis changes everything, not because it instantly creates the modern world, but because it starts a slow revolution with no awareness of itself. Sunlight becomes biology. Biology becomes geology, and waste becomes destiny. The oxygen does not rise like a clean dawn. It creeps, reacts, disappears, returns, and only slowly begins to escape the chemistry that has been swallowing it. For a long time, the planet resists oxygen the way dry soil drinks rain. Iron in the oceans takes it. Volcanic gases take it. Exposed rocks take it. Reduced minerals take it. Every molecule released by oxygen-producing microbes seems to meet some ancient hunger in the Earth itself. the atmosphere remains mostly without free oxygen, and the old anaerobic world continues. But the sinks are not endless. Across immense spans of time, the balance shifts. The planet's chemical appetite begins to weaken, and the waste gas of microbes starts to remain in the air. Around 2.45 billion years ago, though the timing and rhythm remain debated, Earth enters what is known as the Great Oxidation Event. The name sounds sudden, almost like a switch being thrown, but it is better imagined as a long, uneven crossing. Oxygen rises, falls, stabilizes, and interacts with oceans, rocks, climate, and life in ways that are still being reconstructed. Some records suggest pulses rather than a smooth climb. Some environments become oxygenated before others. Deep oceans may remain low in oxygen long after the atmosphere has changed. The evidence remains incomplete, but the broad transformation is unmistakable. Earth is no longer the same planet. A gas once nearly absent from the air becomes a force that changes everything. For many organisms, this is not a blessing. Oxygen is reactive. It damages cells by generating chemical species that attack proteins, membranes, and genetic material. In a world built around anaerobic metabolism, oxygen is not the breath of life. It is a contaminant. To early microbes adapted to oxygen-free environments, rising oxygen may have been one of the greatest environmental crises in Earth history. The phrase oxygen catastrophe captures this from the perspective of the old biosphere. It does not mean every anaerobe vanished. Many survived by retreating into sediments, deep waters, hydrothermal systems, digestive tracts of later animals, and countless oxygen-poor microenvironments. Their descendants still exist today, thriving where oxygen is absent or scarce, but the surface world begins to change against them. At the same time, catastrophe and opportunity are difficult to separate. Oxygen is dangerous, but it also opens a new way to harvest energy. Aerobic respiration, the use of oxygen as a final electron acceptor, can yield far more usable energy from food molecules than many anaerobic pathways. That extra energy does not instantly create animals or complex life, but it expands what is possible. Cells that learn to defend against oxygen, and later to use it, gain access to a powerful metabolism. The same gas that poisons one world becomes the fuel of another. Evolution often works through such reversals. A waste product becomes a resource, a crisis becomes a doorway, a toxin becomes, in time, breath. The mineral record carries the drama in stone. Before widespread oxygenation, certain minerals could survive at the surface that would later be unstable in an oxygen-rich world. After oxygen rises, new weathering patterns appear. The great banded iron formations, built across earlier intervals when oxygen met iron-rich seas, eventually decline as dissolved iron is stripped from large parts of the ocean. Red beds, iron-rich sediments stained by oxidized minerals, become more common later, marking environments where oxygen has changed the chemistry of exposed land. These rocks are not merely backgrounds, they are the planet reacting to life. Microbes alter the air alters the rocks, and the rocks preserve the shock. The oceans become divided in complicated ways. The surface may hold more oxygen, especially where photosynthetic microbes are active, while deeper waters remain anoxic or rich in other chemicals. Some parts of the ocean may become euxinic, low in oxygen and high in sulfide, creating conditions hostile to many forms of later complex life. Scholars still debate the exact pattern of ocean oxygenation, and recent work continues to show that the rise of oxygen was not a single clean step. Earth's atmosphere and oceans oxygenated in stages, with long pauses, reversals, and regional differences. The modern oxygen-rich planet is the end result of a long negotiation, not an immediate consequence of photosynthesis. The climate may also have been shaken. Methane, produced by microbes and stable in an oxygen-poor atmosphere, is a powerful greenhouse gas. As oxygen rises, methane can be destroyed more efficiently, weakening its warming effect. Some scientists connect this atmospheric shift to ancient global glaciations, perhaps even snowball Earth conditions, when ice may have expanded dramatically across the planet. The details remain debated, and no single explanation fully accounts for every climate signal. But the possibility is striking. Microbes releasing oxygen may have helped cool the planet on a catastrophic scale. Life changes the atmosphere changes the climate, and the climate tests life again. In this older world, crises unfold without eyes to witness them. There are no forests freezing under snow, no animals migrating away from cold seas, no human storytellers naming the disaster. The beings most affected are microscopic. They live in films, sediments, waters, pores, and chemical gradients. Their losses leave no bones. Their adaptations leave no dramatic fossils. Yet this invisible crisis may have remade the future more deeply than many later extinctions that seem more spectacular. The oxygen catastrophe does not merely remove some life and favour other life. It changes the basic chemistry of Earth's surface, making possible new metabolisms, new minerals, and eventually new kinds of bodies. One surprising consequence is protection. Oxygen in the atmosphere can form ozone high above the surface, and ozone absorbs much of the sun's ultraviolet radiation. The development of an ozone layer was not immediate in the modern sense, and early oxygen levels remained far below today's for long periods. But even partial changes would have altered the radiation environment. Over time, the surface becomes less hostile to exposed biology. This matters later for life near the surface and, much later, for life on land. Again, a dangerous gas becomes a shield. The transformation is full of such contradictions. Life also evolves defenses. Enzymes that neutralize reactive oxygen species become essential for organisms living near oxygen. Antioxidant systems, repair mechanisms, and new metabolic strategies allow cells to survive in conditions that would once have been lethal. Some microbes become obligate aerobes, dependent on oxygen. Others remain anaerobes, avoiding it. Others become flexible, switching strategies according to conditions. The biosphere diversifies not only through shape, because shape is still mostly microscopic, but through chemistry. The planet becomes a mosaic of metabolic possibilities. The Great Oxidation Event is therefore not simply the moment Earth gains oxygen. It is the moment the relationship between life and planet becomes undeniable. Before this, life has already existed for a long time, perhaps for well over 1 billion years. But now its waste alters the atmosphere itself. No animal has appeared. No plant has rooted in soil. No large body has crossed the seafloor. Yet Earth has entered a new age because microbes have changed the rules. The old anaerobic planet does not vanish completely. It retreats into hidden places and persists. The new oxygenated planet does not arrive fully formed. It grows slowly, unevenly, with long intervals of uncertainty. Between them lies one of the great transitions in deep time, a world poisoned by its own life, and made more powerful by that poisoning. In the quiet aftermath, the stage is different. Oxygen is no longer just a local accident inside microbial mats. It is part of the atmosphere's future. Energy-rich metabolism becomes more available. The chemical barriers that once defined early life begin to shift. Somewhere within this altered world, another strange merger will eventually take place. one that will turn simple cells into something far more internally complex. The oxygen crisis has not produced animals, but it has prepared a planet where complexity can begin to gather itself inside a cell. Complex life begins not with a large body, but with a cell that becomes strange inside. For most of Earth's history, life remains microbial, and much of it is prokaryotic in form small cells without a nucleus, without intricate internal compartments familiar from plants, animals, fungi, and many single-celled eukaryotes. These prokaryotic cells are not failures or unfinished sketches. They are astonishingly successful. Bacteria and archaea survive nearly everywhere life can survive. From boiling springs to deep sediments, from salty lakes to the human gut. If evolution had a preference for simplicity, they would be its masterpiece. But somewhere in the ancient microbial world, one lineage begins to change in a way that opens an entirely new future. It becomes larger, more internally divided, more dependent on partnerships, and eventually capable of giving rise to the cells from which all animals, plants, fungi, algae, and many protists descend. This transition is called eukaryogenesis, the origin of the eukaryotic cell. The word is technical, but the event is almost mythic in importance. A eukaryotic cell has a nucleus, where much of its genetic material is enclosed. It has internal membranes that divide space into working regions. It has a cytoskeleton, a dynamic inner scaffold that helps shape the cell, move materials, and in some lineages, crawl, engulf, or change form. It has elaborate systems for sorting proteins, handling waste, managing signals, and moving energy through specialized structures. Compared with a typical bacterium, a eukaryotic cell can feel like a city inside a wall. Yet it did not appear all at once. The evidence remains incomplete, and scholars still debate the sequence of events. But the broad picture is one of merger, inheritance, and deep microbial collaboration. At the center of this story are mitochondria. In nearly every eukaryotic lineage, mitochondria or their reduced descendants appear as energy-handling organelles. They are the reason many complex cells can support large genomes, dynamic internal structures, and energy-hungry behavior. But mitochondria were not invented from nothing by a host cell. They descend from bacteria, long ago. An archaeal host cell entered into a lasting symbiosis with an alpha proteobacterium-like partner. Whether the host engulfed the bacterium-like prey, sheltered it gradually, or formed a more intimate metabolic partnership before full internalization remains debated. But the result changed life. A bacterium became an organelle. A former outsider became a permanent inner companion. The evidence for this is strong because mitochondria still carry traces of their bacterial ancestry. They have their own DNA, though much reduced. They divide in ways reminiscent of bacteria. Their membranes, ribosomes, and genetic systems reveal deep evolutionary connections to bacterial life. Over time, many genes from the mitochondrial ancestor moved into the host genome, while others were lost. The relationship became irreversible. The host could not easily return to its earlier condition, and the symbiont could no longer live as it once had. This is one of evolution's most remarkable reversals of identity. Two lineages become one organism, not by blending smoothly at first, but through a long process of dependence. The host side of the story has become especially interesting because of Asgard archaea. These microbes, first recognized from genetic evidence in sediments and later studied in more detail, are archaea whose genes show surprising relationships to eukaryotic cell biology. Some Asgard lineages carry versions of genes connected to cytoskeleton-like systems, membrane remodeling, and other features once thought to belong mainly to eukaryotes. Recent research has strengthened the idea that eukaryotes emerge from within, or very near, the Asgard archaeal branch, with contributions from bacterial partners and other microbial interactions. This does not mean modern Asgard archaea are our direct ancestors. They are living relatives of a broader ancient lineage, changed by their own evolution. But they help illuminate the kind of host that may have been capable of taking the first steps toward eukaryotic complexity. The world in which this merger occurred was probably still mostly microbial and chemically layered. Oxygen existed, but not necessarily at modern levels. Many environments remained low in oxygen, while others experienced more oxygenated conditions. This matters because mitochondria are tied to energy metabolism that can use oxygen efficiently, though the earlier symbiosis may have involved more complicated exchanges involving hydrogen, carbon compounds, sulfur, or other metabolic currencies. No single explanation fully accounts for the first partnership. Some models imagine the symbiont helping the host deal with oxygen. Others emphasize hydrogen transfer or syntrophic relationships, where one organism lives from the waste products of another. What all these models share is the idea that eukaryotic complexity did not arise from isolation. It arose from intimacy. This is a profound shift in the story of evolution. Earlier life changes the planet through metabolism and photosynthesis. Now life changes itself by bringing another life form inside. Evolution is often pictured as competition, and competition is real, but the origin of complex cells reminds us that cooperation can be just as radical. The mitochondrion is not a polite alliance between neighbors. It is a captured partnership so deep that the partners become inseparable. Later, in the lineage leading to plants and algae, another major symbiosis will occur when a eukaryotic cell incorporates a photosynthetic cyanobacterium-like organism, giving rise to chloroplasts. The green world of leaves and forests ultimately depends on that second great internal merger. The first merger makes complex eukaryotic cells possible. The second gives some of them sunlight. Inside early eukaryotes, the nucleus becomes another mystery. Why did the nuclear membrane evolve? How did internal membranes develop? Did they arise from folds of the host membrane, from vesicles, from interactions with symbionts, or through a combination of processes? The answers remain debated, but the nucleus changes the relationship between genes and the rest of the cell. It separates transcription from translation, allowing new layers of regulation, introns, splicing. Chromosome organization and complex gene control become increasingly important. This does not automatically produce large organisms, but it gives cells new ways to manage information. Complexity begins as control over timing, location, and interaction inside a single cell. This cytoskeleton adds movement and form. With internal filaments and motors, eukaryotic cells can move materials through their interiors, change shape, engulf particles, divide chromosomes in organized ways, and eventually form the basis for cell cooperation in multicellular bodies. Some single-celled eukaryotes become predators, consuming bacteria and other small cells. This changes microbial ecosystems. For the first time, cells can hunt in a more physically elaborate sense. Not merely absorb dissolved molecules or chemical products. Predation may have encouraged defenses, larger size, armor-like coverings, faster movement, and more complex interactions. Even before animals, the microscopic world becomes an arena of pursuit and escape. For a long time, eukaryotes remain mostly single-celled. They diversify in oceans and sediments, in planktonic worlds and microbial communities that leave only scattered chemical and fossil hints. Their early record is difficult because soft single cells preserve poorly, and many ancient microfossils are hard to classify. Some large organic walled fossils may represent early eukaryotes, but certainty is often hard-won. What survives in the record suggests that eukaryotes emerged well before animals, perhaps more than 1.5 billion years ago, and then spent immense spans of time experimenting at small scales. The rise of complex cells is not immediately followed by the rise of complex bodies. Evolution often opens a door long before anything walks through it. Still, the door is open, with mitochondria providing energy, internal membranes creating compartments, a nucleus reshaping genetic control, and a cytoskeleton allowing movement and structure. Life has changed its architecture. The planet is still ruled by microbes, and in many ways it always will be. But within that microbial kingdom, a new kind of cell now exists, one capable of becoming many things. It can be predator, symbiont, photosynthesizer, drifter, builder, parasite, partner. It can join with others. It can specialize. It can begin the long experiment that will eventually lead to bodies, tissues, senses, and the visible life of later seas. The great leap toward animals does not begin with an animal. It begins with a cell that carries another cell inside itself, a living memory of partnership burning quietly at its center. Once complex cells exist, the next great transformation is not inevitable. A eukaryotic cell can be intricate, energetic, predatory, photosynthetic, and mobile without ever becoming part of a body. For hundreds of millions of years, much of eukaryotic life remains single-celled or loosely colonial, thriving in ways that do not require tissues, organs, or permanent cooperation. This is easy to overlook because the modern world is crowded with bodies, trees, insects, fish, birds, mammals, mushrooms, seaweeds, and flowers make multicellularity seem like the obvious direction of evolution. But for most of life's history, the successful strategy is still to be small. A single cell can feed, divide, drift, hunt, hide, and adapt without negotiating with neighbors. To become multicellular, cells must give up some independence, and in evolution, surrendering independence only works when the bargain pays. The simplest version of multicellularity is not strange at all. Cells often remain together after division. Microbes form mats, filaments, sheets, clusters, and colonies. Bacteria did this long before animals existed. Some cyanobacteria form chains in which certain cells specialize for nitrogen fixation. Some algae form colonies where cells coordinate movement. Slime molds can gather into temporary collective forms when conditions demand it. Even today, the boundary between a colony and a true multicellular organism is not always as sharp as textbooks make it seem. The evidence remains incomplete for the deep past, but modern life shows that cooperation among cells has evolved many times. The hard step is not simply sticking together, it is making the togetherness stable, regulated, inherited, and useful. A true multicellular body requires trust among cells, though not trust in any conscious sense. It requires rules. Some cells must specialize, some may reproduce, while others support reproduction without passing on their own lineage directly. Some must die at the right time to shape tissues or protect the organism. Some must stay in place. Some must obey signals. If cells begin competing selfishly inside the body fails. Cancer in modern animals is a reminder that multicellularity is always a negotiated peace. The ancient origin of bodies therefore required more than physical attachment. It required genetic and chemical systems that could coordinate behavior, control growth, manage conflict, and make a whole organism more successful than a crowd of separate cells. The first experiments with multicellularity probably happened many times, in many lineages, most leaving little trace. Plants, animals, fungi, red algae, brown algae, green algae, and several other groups all reached multicellular forms independently. This repeated evolution suggests that once eukaryotic cells had enough internal complexity, multicellularity became a tempting possibility under the right conditions. Larger size could protect against predators. A cluster might be harder to swallow than a single cell. Division of labor could make feeding, movement, or reproduction more efficient. A body could lift photosynthetic cells toward light or anchor feeding cells in a favorable current, but no single explanation fully accounts for why some lineages built bodies while others remained single cells. Evolution is not a ladder, it is a landscape of trade-offs. Oxygen may have mattered, but again, not as a simple switch. Larger bodies often require better energy supply and, eventually, ways to move oxygen or nutrients through tissues. Rising oxygen levels in the oceans and atmosphere may have expanded the possibilities for bigger, more active life. Yet early multicellularity appears in a world where oxygen conditions vary across time and place. Some forms may have grown slowly in low-energy settings. Others may have depended on oxygenated surface waters. Scholars still debate how closely the rise of animals was tied to changing oxygen, ecological competition, genetic innovation, or all of these together. What survives in the record suggests that the path to bodies was not one invention, but a convergence of many conditions: energy, genes, environment, predation, development, and time. Before animals become obvious in fossils, there are quieter hints: organic molecules, microfossils, strange impressions, and possible traces of early multicellular algae suggest that complex life was experimenting long before the seafloor filled with shells and burrows. Some ancient fossils appear as simple filaments or sheets. Others are difficult to classify, hovering between microbial colonies, algae, and early eukaryotic experiments. The problem is that soft bodies are poor at leaving evidence. A jelly-like organism can live, feed, reproduce, and disappear without making a fossil that survives metamorphism and burial. The early history of multicellularity may therefore be much richer than the rocks reveal. The silence is not absence. Sometimes it is poor preservation. One lesser-known detail is that many of the genes needed for animal bodies have roots older than animals themselves. Single-celled relatives of animals possess versions of genes involved in adhesion, signaling, and coordination. They do not have animal tissues, but they have molecular tools that animals later use to build tissues. This suggests that evolution often repurposes old parts for new architecture. The first animals did not need every bodybuilding gene to appear from nowhere. Some of the toolkit already existed for interactions between single cells and their environments, for sensing, attaching, communicating, or responding. Multicellularity is partly the story of old cellular habits becoming internal politics. At the scale of daily life, if daily can even be imagined across such ancient oceans, the world before animals is not empty. It is full of films, mats, drifting cells, colonies, algae-like forms, predators too small to see, and chemical gradients. The seafloor may be covered in microbial carpets in many places, stable surfaces where cells trap sediment and create layered textures. In the water column, tiny eukaryotes drift, feed, divide, and are eaten. Some cells engulf others. Some use sunlight, some live by partnership, some perhaps gather into loose groups, then tighter groups, then inherited bodies. The world is alive, but its life is still soft, thin, and easily erased. The emergence of development is one of the great hidden revolutions. A multicellular organism is not merely a mass of cells. It begins from a single cell, or a small starting structure, and produces a patterned form through regulated division, movement, and differentiation. This requires cells to know, chemically, where they are in relation to one another, front and back, inside and outside, surface and interior, reproductive and supportive tissues. These distinctions do not arise by accident in each generation. They are built through developmental programs. Early versions were probably far simpler than those in modern animals, but even simple patterning changes everything. It allows bodies to become repeatable. Repeatability gives evolution something new to refine. A body plan can be inherited, modified, enlarged, simplified, or specialized. A sheet can become folded, a hollow ball can become layered, cells on the outside can handle contact with the environment, while cells inside take on other roles. In animals, this will eventually lead to tissues, guts, muscles, nerves, and senses. But at this stage, the story should not rush ahead. The first multicellular organisms do not know they are preparing the Cambrian. They are only surviving. They are solving local problems: how to stay together, how to feed, how to grow without tearing apart, how to reproduce without losing organization, how to keep internal conflict under control. The quiet prelude before visible animals dominates the fossil record may have lasted hundreds of millions of years. This long delay is important. It shows that complexity does not explode the moment it becomes possible. The planet must be ready. Ecosystems must shift. Genetic systems must accumulate. And bodies must be tested in forms that mostly vanish. Evolution spends immense time in preparation, though preparation implies a destination, and there was none. There were only lineages continuing, failing, merging, adapting, and occasionally discovering a form of cooperation stable enough to persist. By the end of this long cellular experiment, life has acquired a new scale. It is still microbial at its foundation, still dependent on chemistry older than oxygen, older than nuclei, older than bodies. But now cells can become architecture. They can arrange themselves into shapes larger than a single cell's experience. The ancient ocean begins to hold organisms that are not yet familiar animals, not yet the armoured and burrowing creatures of later seas, but soft experiments in form. The next world will seem dreamlike because its inhabitants do not fit easily into modern categories. They will appear as fronds, discs, quilted shapes, tubes, and impressions pressed into ancient sediment, as if life is rehearsing bodies before it decides what a body can be. The first large soft bodies appear like ghosts pressed into stone. They do not arrive with jaws, eyes, legs, shells, or bones. They do not look like the animals that later fill the seas. Many have no obvious head, no clear mouth, no visible organs, and no easy place in the familiar tree of life. They spread across ancient seafloors as quilted ovals, ribbed fronds, branching forms, tubes, discs, and strange leaf-like shapes anchored in soft sediment. They belong to the Ediacaran world, a period that feels less like the opening act of modern animal life and more like a dream Earth once had before waking into the sharper, more violent geometry of the Cambrian. Yet this dream is not imaginary. It is fossilized in delicate impressions, preserved where soft bodies met microbial mats and fine sediment settled over them with unusual gentleness. By roughly 635 million years ago, Earth is emerging from severe climatic episodes that may have covered large parts of the planet in ice. The details of these glaciations remain debated, but the aftermath matters. Oceans change, nutrients shift. Oxygen levels may rise in some environments. Continental margins host broad, shallow seas. Microbial mats still carpet large areas of the seafloor, creating firm, textured surfaces unlike the churned sediments of later animal-dominated oceans. On these mat grounds, soft organisms can rest, feed, grow, and leave impressions. Without widespread burrowing animals to disturb the sediment, their traces can survive in ways that later soft-bodied life often cannot. The Ediacaran fossil record is therefore not only a record of organisms, it is a record of a world before the seafloor was fully disturbed by restless bodies. Dickinsonia is one of the most famous of these forms. It appears as an oval, ribbed body, sometimes small, sometimes over a metre long, with a quilted pattern that seems symmetrical, but not quite like most later animals. For decades, scientists debated what Dickinsonia was: an animal, a fungus-like organism, a giant protist, a lichen-like form, or something belonging to an extinct experiment in multicellular life. Chemical evidence from some fossils has strengthened the case that Dickinsonia was an animal, or very close to animals, but its exact way of life remains uncertain. It may have fed by absorbing nutrients from microbial mats beneath it, leaving faint traces as it moved or settled. If so, it was not a hunter in any dramatic sense. It was a quiet grazer of microbial landscapes, a soft body living from the skin of the sea floor. Other Ediacaran organisms complicate the picture further. Frond-like forms such as Charnia rise from the sediment in branching patterns, looking almost plant-like, though they lived in the sea and were not plants. Some may have absorbed nutrients directly from seawater. Funesia, a tube-like organism known from ancient deposits, may represent one of the earlier examples of animals with sexual reproduction, inferred from patterns in fossil populations, though interpretations remain cautious. Cambrian, found in Ediacaran rocks, looks more recognisably animal-like to many researchers, with evidence suggesting it may have had a muscular underside and perhaps scraped microbial mats with a feeding structure. If that interpretation is correct, Cambrian begins to feel like a creature leaning toward the later world, still soft, still strange, but more active, more directed, more involved in changing the surface it lives on. In northwestern Canada, Ediacaran fossil sites preserve a striking diversity of these ancient forms around 567 million years ago, including organisms such as Dickinsonia, Funesia, and Cambrian-like traces in ecosystems more complex than old, simple narratives allowed. This matters because the Ediacaran is no longer seen merely as an empty waiting room before the Cambrian explosion. It is a living world in its own right, full of ecological experiments. Some organisms are anchored, some lie flat. Some may move, some build tubes or appear in repeated clusters. Some leave trails, suggesting behaviour rather than passive existence. The evidence remains incomplete, but what survives suggests that complex life was already diversifying before the sudden abundance of hard parts makes the Cambrian fossil record seem so dramatic. The atmosphere and oceans are changing during this time, but not in a simple upward line. Oxygen likely rises in stages, and different marine settings may contain very different amounts of it. Some Ediacaran organisms may have lived in oxygenated shallow waters, while other environments remained chemically challenging. Larger bodies require oxygen not merely to exist but to move, grow, and maintain tissues. Yet these first large organisms may have used forms and lifestyles that minimize those demands. Thin, broad bodies increase surface area. Fronds expose more tissue to surrounding water. Slow growth and passive feeding reduce energy needs. Life is testing geometry as much as it is testing biology. One of the most beautiful and unsettling things about Ediacaran life is how much of it disappears. Many iconic forms do not seem to pass clearly into the Cambrian world. Some may have descendants hidden among later animals, but others may represent extinct body plans, evolutionary experiments that flourished under Ediacaran conditions and then faded when those conditions changed. The microbial mat grounds that supported them begin to be disturbed by more active animals. Burrowing increases. Predation becomes more important. Skeletons and shells alter ecological relationships. The soft quilted forms of the Ediacaran may have been well suited to a quiet seafloor, but poorly suited to a world of digging, biting, hiding, and chasing. No single explanation fully accounts for their decline, and scholars still debate whether they were outcompeted, eaten, environmentally stressed, or simply transformed into lineages harder to recognize. The Ediacaran also challenges the idea that evolution always moves from simple to complex in a direct way. Some forms are large but not obviously complex in the same sense as later animals. Some may have had no true gut, no nerves, perhaps no organs as we understand them. Others show hints of more familiar animal organization. Complexity appears unevenly. Size, movement, feeding, development, and tissue organization do not all advance together. Evolution is experimenting with combinations. A body can be large and passive. A smaller organism can be more behaviorally active. A simple-looking fossil can hide a sophisticated developmental pattern. A strange form can be successful for millions of years and still leave no lasting branch. For a long-form story of life on Earth, this world is a necessary pause. It resists the temptation to rush from microbes to animals as though the path were obvious. Here, the planet is alive with bodies, but the bodies do not yet belong to the world we know. They are soft, flat, quilted, anchored, drifting, grazing, absorbing, perhaps moving slowly over microbial films beneath dim, ancient water. They live before eyes become common weapons, before shells become widespread armor, before burrows turn the seafloor inside out. They are the first large shadows of animal possibility, and yet many of them remain unnamed by any modern category. When the Ediacaran seafloor is imagined at night, if night can be used for a world without human watchers, it is not empty. It is a quilt of living mats and soft forms resting in stillness. Fronds stand in gentle currents. Oval bodies lie on textured surfaces. Tiny traces cross the sediment where something has moved with purpose. Chemistry, oxygen, genes, and ecology are all converging, but not toward a guaranteed outcome. The future is nearby, though nothing here knows it. The Cambrian Sea is approaching, with sharper bodies and more urgent lives. But before that world arrives, Earth holds this quieter one, a dream world of soft experiments, preserved only because mud, microbes, and chance once covered them gently enough to let their shapes remain. The Cambrian Sea does not begin as an explosion in the ordinary sense. There is no single instant when the ocean suddenly fills with eyes, claws, shells, spines, and swimming bodies. The name Cambrian explosion is a human phrase, made from the shock of the fossil record, where after long ages of microbial films and soft impressions, animal life appears with a new sharpness. Skeletons harden, burrows cut through sediment, predators leave damage on shells. Eyes begin to change the meaning of distance. Bodies develop fronts and backs, tops and bottoms, mouths, guts, limbs, armor. and ways of moving that make the seafloor feel restless. It is not life beginning. It is life becoming visible in a new way. The transition begins after the Ediacaran world has already shown that large complex organisms exist. But Cambrian fossils reveal something different: ecological intensity. Animals are no longer mostly soft forms resting on microbial mats or moving slowly across quiet surfaces. They are digging, crawling, swimming, filtering, grazing, grabbing, hiding, and defending themselves. The sea floor, once stabilized by microbial carpets, becomes churned by burrows and tracks. This change is sometimes called the agronomic revolution, a strange term for an ancient ocean, but it captures the scale of disturbance. Animals begin ploughing through sediment, mixing it, aerating it, breaking up mat grounds, and creating a new three-dimensional world beneath the surface. The ground itself becomes an ecosystem. The fossil sites that make the Cambrian feel so alive are often places of exceptional preservation. The Burgess Shale in Canada, the Chengjiang biota in China, and other deposits reveal soft tissues that would normally disappear. They show not only shells and hard parts, but limbs, guts, eyes, gills, delicate appendages, and bodies too strange to classify easily when they were first discovered. Without such sites, the Cambrian would look much poorer and simpler. With them, it becomes a crowded ancient theatre. There are worm-like animals with grasping spines, early arthropods with jointed limbs, sponge-like forms, lobopodians with soft legs, early chordates that hint faintly toward the distant ancestry of vertebrates, and filter feeders arranged like living baskets in the water. Some creatures seem familiar in outline. Others look like evolution tried a design and never repeated it quite the same way again. Anomalocaris is one of the classic symbols of this world. For a long time, parts of its body were mistaken for separate animals grasping appendages interpreted as shrimp-like creatures, a circular mouth as something jellyfish-like, other fragments as unrelated forms. Only later did these pieces come together into one large swimming predator, with frontal appendages, a ring-like mouth, side flaps, and prominent eyes. It was not a monster in the exaggerated sense, but for its time it was a major active hunter, a sign that predation had become an organizing force. Once animals can chase, grasp, crush, pierce, or bite, other animals face new pressures. Armor becomes useful. Burrowing becomes useful. Speed becomes useful. Senses become useful. The Cambrian Sea begins to run on pursuit and avoidance. Eyes may be one of the most important inventions in this shift. Light-sensitive cells existed before complex eyes, but the Cambrian sees the rise of visual systems capable of detecting direction, shape, movement, and perhaps increasingly detailed images. Trilobites, among the most famous Cambrian arthropods, possess compound eyes made with calcite lenses in many species. Vision changes behavior because it extends the world beyond touch and chemistry. A sighted animal can detect prey or danger before contact. It can aim movement. It can choose direction with more precision. The ocean becomes not only a chemical landscape, but a visible one. Once vision appears, it pressures others to hide, harden, flee, or deceive. A new kind of arms race begins, though scholars still debate how central vision was compared with oxygen, genetics, ecology, and environmental change. The causes of the Cambrian diversification remain debated because no single explanation fully accounts for its timing and scale. Oxygen almost certainly matters, but not as a simple magic ingredient. Larger and more active bodies require more energy, and oxygen-rich metabolism helps supply it, yet oxygen levels likely varied widely by environment. Developmental genes matter too. The genetic toolkit that helps build animal bodies, including regulatory genes that pattern form, may have deep roots before the Cambrian, but the way those genes were deployed could produce new anatomical possibilities. Ecology matters, predation, burrowing, competition, and new food webs can accelerate innovation. Environment matters, changing seas, continents, nutrients, and ocean chemistry may have opened new habitats. The Cambrian explosion is less a single cause than a convergence, a moment when many pressures and possibilities meet. Hard parts are especially important to the fossil record. Shells, spines, plates, and mineralized skeletons preserve more readily than soft tissue, so their spread makes animals more visible to geology. Mineralization may have evolved for defense, support, feeding, or mineral regulation, and perhaps for different reasons in different lineages. Once hard parts exist, they reshape ecosystems. A shell protects, but it is also weight. A spine deters, but it costs material and energy. A burrow protects, but it requires movement and changes feeding. Every innovation comes with trade-offs. The Cambrian world is not simply becoming better, it is becoming more entangled. The animals themselves are still marine. Land remains mostly barren, at least compared with later ages, though microbial life may exist in damp terrestrial settings. The great drama is underwater, on continental shelves and shallow seas where sunlight, nutrients, oxygen, and sediment create habitats dense with activity. Microbial mats do not vanish everywhere, but they lose their old dominance in many settings. Grazers scrape them, burrowers disrupt them. Filter feeders pull particles from the water. Predators patrol above or within the sediment. The quiet Ediacaran surface gives way to a world where the boundary between water and seabed is constantly being worked. One lesser-known detail is that the Cambrian does not only create modern-looking groups, it also preserves lineages and body plans that are difficult to place. Hallucigenia, once reconstructed upside down and backward, shows how strange these animals can appear to modern eyes. Opabenia, with its 5 eyes and flexible frontal appendage, once seemed almost absurd. Wiwaxia carries scales and spines but has affinities that are still discussed. These fossils remind us that early animal evolution was not a clean parade of ancestors marching toward familiar forms. It was an experimental radiation, with many branches flourishing briefly or persistently before some faded from the record. The future did not belong automatically to the forms that look most normal to us now. Still, the foundations of later animal life are being laid. Arthropods diversify and will become one of the most successful groups in Earth history. Mollusk-like lineages begin long journeys toward shells, mussels, and later forms of movement. Worm-like animals reshape sediments. Early chordates carry features that, far in the future, will be part of the vertebrate story, a body axis, a notochord-like structure, and a new way of organizing movement. These beginnings should not be treated as tiny humans waiting to happen. They are their own organisms, living in their own seas, solving their own problems, but from the deep distance of time, they show that the architecture of later ecosystems is appearing predator and prey, swimmer and burrower, grazer and mat, shell and tooth, eye and shadow. The Cambrian explosion marks a change in visibility, behavior, and ecological complexity. Life has moved from chemistry into cells, from cells into symbiosis, from symbiosis into bodies, and now from soft bodies into active ecosystems rich with conflict, defense, movement, and sensation. The planet is still ancient beyond ordinary imagination, and humans remain unimaginably far away. But after the Cambrian, Earth is no longer merely a microbial planet with occasional soft experiments. It is a planet of animals, and the ocean has become a place where life does not only endure, it searches, escapes, builds, breaks, hides, hardens, and watches. For almost all of life's history so far, the great story has belonged to water, chemistry gathered in water, cells endured in water, photosynthesis transformed water-lit surfaces, animals diversified in water, and the Cambrian world sharpened itself beneath ancient seas. Land, by comparison, remained a harsher frontier. It was not empty in every sense, because microbes could live on damp rock, in films, soils in the making, and perhaps in sheltered patches where moisture lingered. But it was not yet a green world. There were no forests catching mist, no roots gripping hillsides, no insects moving through leaf litter, no vertebrate footprints pressed beside rivers. The continents were vast exposed surfaces of stone, sediment, wind, ultraviolet light, heat, cold, and chemical weathering. To life shaped by oceans, land was both opportunity and danger. The first conquest of land was not a dramatic march of animals from the sea. It was slower, thinner, and quieter. Microbes came first, or perhaps never truly stayed away wherever moisture allowed them to persist. They formed crusts on rock and sediment, broke minerals down, trapped particles, and helped create the earliest hints of soil. Fungi or fungus-like organisms may have played important roles in this transition, partnering with early plants or plant-like algae, digesting rock, moving nutrients, and weaving through damp substrates. The evidence remains incomplete because early terrestrial life was small, soft, and easily erased. But what survives in the record suggests that land became habitable through cooperation long before it became visibly lush. Life did not simply step onto a ready-made continent. It helped manufacture the continent's living skin. Plants, when they begin to establish themselves on land, face problems that sea life never had to solve in quite the same way. In water, buoyancy supports bodies. Reproduction can use swimming cells. Drying is rarely the central threat. Nutrients are dissolved around the organism. On land, a plant must avoid desiccation, hold itself upright, exchange gases without losing too much water, protect reproductive cells, and draw minerals from surfaces that are not yet rich soil. Early land plants were probably small, simple, and closely tied to damp environments. They did not resemble towering trees. They were more like low green films, mats, branching axes, and simple stems hugging wet ground. Yet their arrival changes the planet. Even small plants trap sediment, slow erosion, alter rivers, deepen weathering, and feed carbon into the ground. The partnership between plants and fungi may be one of the great underappreciated engines of Earth history. Many modern plants depend on mycorrhizal fungi, which extend through soil and help gather minerals, especially phosphorus, while receiving carbon compounds from the plant. Fossil and molecular evidence suggests that such partnerships, or ancestral versions of them, are ancient. This matters because early plants trying to live on bare mineral surfaces would have needed help. Fungi are chemical miners. They can dissolve minerals, explore tiny spaces, and connect living tissues to nutrients beyond their immediate reach. The greening of land may therefore have been less a conquest by plants alone than a negotiated alliance between kingdoms of life. As with mitochondria inside early eukaryotes, cooperation again changes destiny. As vegetation spreads, the atmosphere and climate respond. Plants draw carbon dioxide from the air through photosynthesis and lock carbon into tissues, sediments, and soils. Roots and fungal networks accelerate weathering, which can also remove carbon dioxide over long timescales. Rivers begin carrying new kinds of organic material to the sea. Soils thicken. Landscapes stabilize in some places and become more complex in others. The continents stop being merely geology exposed to air and begin becoming ecosystems. This transformation is gradual, uneven, and debated in its details, but its importance is enormous. Life moves onto land and then changes the land's chemistry, structure, and climate, just as microbes once changed the atmosphere. Arthropods are among the first animals to exploit these new terrestrial spaces. Their hard exoskeletons help protect them from drying and injury. Jointed legs allow movement across uneven surfaces. Some may feed on microbial films, decaying plant material, spores, or one another. Millipede-like animals, early arachnids, and insect relatives eventually enter terrestrial ecosystems, though the exact sequence is complex and still debated. To imagine these first land animals, it is better not to picture a crowded forest floor at first, but damp margins, riverbanks, low vegetation, fungal mats, and patchy habitats where small bodies move between shelter and exposure. The first terrestrial animal worlds were likely fragile, moisture-bound, and seasonal. The land could feed them, but it could also dry them out. Plants respond to land's challenges with structure. Vascular tissue allows water and nutrients to move through taller bodies. Waxy cuticles reduce water loss. Stomata regulate gas exchange. Spores and later seeds protect reproduction from the need for constant free water. Lignin strengthens tissues, allowing plants to rise higher. With height comes competition for light, and with competition for light comes the architecture of forests. By the Devonian and Carboniferous periods, immense plant communities reshape lowlands and wetlands. Forests form canopies, roots penetrate deeply. Dead plant material accumulates in waterlogged swamps, sometimes escaping complete decay and eventually contributing to coal deposits. The air itself changes as photosynthesis, burial, and decomposition interact across millions of years. These ancient forests are not modern forests. Many are built from lycopsids, horsetail relatives, seed ferns, and early trees with forms that would feel unfamiliar today. Some tower over swampy ground in humid lowlands. Giant club moss relatives rise like columns with crowns of narrow leaves. The forest floor may be wet, dim, and thick with decaying vegetation. Insects and other arthropods diversify in the shelter of plants. Some grow large in oxygen-rich periods. Though the relationship between oxygen and arthropod size is not the only factor, still, the Carboniferous world reminds us that life on land quickly becomes atmospheric, ecological, and geological all at once. A forest is not just a collection of plants. It is a machine that changes air, water, soil, and future rock. Vertebrates arrive on land later, not as triumphant conquerors, but as animals already tied to shallow waters, wetlands, and marginal habitats. Their ancestors were lobe-finned fishes with strong fins, lungs or lung-like organs in some lineages, and bodies suited to complex aquatic environments. The transition from fish-like animals to early tetrapods is not a simple leap from sea to shore. It unfolds through swampy, shallow, plant-filled landscapes where moving through tangled water, breathing air, or pushing across mud could be useful. Fossils such as Tiktaalik and other early tetrapod relatives reveal mixtures of traits, fish-like features alongside necks, limb bones, wrists, and skull changes that hint at life near the water's edge. Scholars still debate the exact ecological pressures, but the transition was gradual, not a single heroic crawl onto dry land. Once vertebrates establish themselves on land, the terrestrial world gains new kinds of bodies and behaviours. Amphibian-like animals remain tied to water for reproduction, but later amniotes evolve eggs and reproductive strategies that free them more fully from aquatic nurseries. Reptile lineage animals diversify. Synapsids, the distant lineage that will eventually include mammals, appear. Ecosystems become layered with plants, arthropods, fungi, amphibians, reptiles, and countless microbes continuing the foundational work beneath all visible life. The conquest of land is not a final victory over water. Every land organism still carries water inside, protects it, moves it, loses it, seeks it, and depends on cycles that return it. By the time land becomes green and inhabited, the 4 billion year story has changed scale again. Life began as chemistry in a world of water and rock. It became cells, then communities, then oxygen, then complex cells, then bodies in the sea. On land, life becomes landscape. It builds soil from stone, makes forests from air and sunlight, turns swamps into future coal, gives rivers roots to cut around, and creates habitats where animals can breathe above the surface that once confined their ancestors. The continents, once hostile and bare, begin to remember life not as faint chemical traces, but as trunks, tracks, spores, burrows, wings, footprints, and the dark buried remains of vanished forests. Life reaches land, fills seas, thickens forests, and builds ecosystems so intricate that they can begin to feel permanent. But permanence is one of deep time's most convincing illusions. Again and again, Earth changes the rules. Oceans lose oxygen. Volcanoes flood continents with lava. Climates swing toward heat or ice. Sea levels rise and fall across continental shelves. Asteroids strike from the dark beyond the atmosphere. Carbon cycles shift. Methane escapes. Acid spreads through seas. Forests collapse. Reefs vanish. Food webs break apart. The story of life is not a smooth climb from simple beginnings to increasing abundance. It is a long survival through repeated ruin, where every flourishing world exists only until the planet, or space, or life itself, changes the conditions beneath it. Extinction is often imagined as an ending, and for the organisms caught inside it, that is exactly what it is. A lineage that disappears has no private consolation in future recovery. It is gone. But at the scale of Earth history, extinction also clears, rearranges, and redirects. This does not make it gentle. It does not make catastrophe purposeful. It means that evolution continues through loss as well as invention. The living world we know is shaped not only by what life created, but by what vanished before it. Many familiar groups exist because other groups were removed, reduced, or pushed aside by crises no creature could understand while they were happening. The first great disruptions are microbial and chemical, and they arrive long before forests or animals. The rise of oxygen itself may have been a catastrophe for anaerobic life, even while it opened the way for energy-rich metabolism. Later, as complex ecosystems develop, extinction becomes easier to see in fossils. Shells disappear from strata, reef communities collapse, trilobites decline and then vanish. Forest types change. Whole marine communities are replaced by others. The fossil record is not perfect, and scholars still debate the exact tempo of many extinction events. But the pattern is unmistakable. Life diversifies, stabilizes for a time, and then something shifts so strongly that survival becomes selective in brutal new ways. The Big Five mass extinctions are the most famous markers, though they are not the only crises. At the end of the Ordovician, around 444 million years ago, major climate change and glaciation are linked to severe marine losses. At the end of the Devonian, a drawn-out series of crises affects reefs, fishes, and marine ecosystems in ways still not fully explained. At the end of the Permian, around 252 million years ago, life comes closest to a global biological collapse. At the end of the Triassic, another extinction opens ecological space before dinosaurs become dominant on land. At the end of the Cretaceous, around 66 million years ago, an asteroid impact helps end the age of non-avian dinosaurs and many marine groups, while birds, mammals, crocodilians, turtles, plants, insects, and countless smaller forms survive into a changed world. The Permian extinction is the darkest of these chapters. Long before the asteroid that ends the Cretaceous, Earth nearly loses the complexity it has spent hundreds of millions of years building. The likely main driver is immense volcanism in Siberia, where eruptions release huge quantities of carbon dioxide and other gases over vast spans of time. The climate warms sharply. Oceans become stressed, acidic, and low in oxygen. Marine ecosystems suffer catastrophic losses. On land, forests and animal communities are transformed. The exact chain of cause and effect remains debated in detail, because the event involves climate, volcanism, ocean chemistry, carbon release, methane, anoxia, and ecological collapse all interacting, but no single image captures it fully. It is not only fire, it is heat, suffocation, acidification, starvation, and the unravelling of systems that had seemed stable. Yet even there, life does not end. Microbes persist, some plants survive, some animals endure in reduced and altered forms. Smallness, flexibility, broad diets, hidden habitats, fast reproduction, or simple luck may help certain lineages cross the crisis boundary. Survival is not always about superiority. Sometimes it is about being in the right refuge, needing less, reproducing quickly, tolerating extremes, or belonging to a lineage whose weaknesses happen not to matter in that particular disaster. Extinction exposes the limits of adaptation. It also exposes the role of chance. The Cretaceous ending impact offers a different kind of catastrophe, faster and more cinematic. An asteroid strikes near what is now the Yucatan Peninsula, blasting dust, sulfur, and debris into the atmosphere. Wildfires may occur in many regions. Sunlight is reduced. Photosynthesis falters. Food chains strain from the bottom upward. Marine plankton communities are hit hard. Non-avian dinosaurs disappear, along with ammonites, many marine reptiles, and numerous other groups. The details of timing, regional survival, and the role of volcanic activity in India's Deccan Traps remain debated, but the impact is one of the clearest examples of a sudden external shock changing evolutionary history. In a geological instant, a world ruled by dinosaurs becomes a world where mammals have room to diversify. This does not mean mammals were waiting heroically for their chance. They had already existed for a very long time alongside dinosaurs, often small, varied, and ecologically clever. After the extinction, some lineages expand into open niches. Over millions of years, mammals become larger, more diverse, and more ecologically prominent. Birds, the living descendants of theropod dinosaurs, also diversify. Forests recover, reefs rebuild in new forms. The world does not return to what it was. Recovery is not restoration, it is reinvention. That is the hidden rhythm after every mass extinction. The biosphere does not simply heal like a wound returning skin to its original shape. It grows into something different. After the Permian, new reptile lineages, marine ecosystems, and eventually dinosaurs rise in a world reshaped by loss. After the Cretaceous, mammals and birds radiate into ecological spaces once occupied by other animals. After reef collapses, new reef builders eventually emerge. After forests disappear, new plant communities form. Life's resilience is real, but it is not sentimental. It preserves continuity by allowing old worlds to die. The lesser-known survivors may matter as much as the famous victims. Burrowing organisms survive where surface conditions fail. Freshwater habitats can become refuges when marine systems collapse. Seeds, spores, cysts, and dormant states allow life to wait through bad intervals. Microbes continue beneath the drama, recycling nutrients, driving decay, stabilizing or destabilizing chemistry. In many crises, the future may depend less on the largest creatures than on small, hidden systems that keep basic biological processes alive. The ancient lesson repeats from the earliest Earth survival often belongs to the quiet forms. Extinctions also reveal that life and planet remain locked together. Life changes climate through carbon burial and gas exchange. Plants alter weathering and oxygen. Microbes transform oceans. Animals disturb sediments. But Earth answers with volcanism, tectonics, orbital shifts, sea level change, and impacts from beyond. Evolution takes place inside this conversation, not above it. A lineage succeeds only under conditions that can change without warning. The stability of any ecosystem is temporary, even if it lasts 10 million years. By the time humans appear far later, they inherit not a perfect ladder of progress, but a world built from survivors of disaster. Every living organism is descended from lineages that crossed earlier bottlenecks. Every body carries the long memory of extinctions it did not personally experience, but would not exist without. The forests, reefs, grasslands, insects, birds, mammals, fungi, and microbes of the present are all late arrangements, assembled after countless vanished worlds. Life began as fragile chemistry, became cells, transformed the atmosphere, built bodies, conquered land, and then endured repeated collapse. Its history is not a straight path upward. It is a pattern of persistence after erasure, a 4 billion year habit of beginning again without ever truly returning to the beginning. Human beings enter this story so late that, if the 4 billion year history of life were a long night, the first humans would appear only in the last faint breath before dawn. By then, nearly everything that makes human life possible has already happened. The planet has cooled from its violent youth, oceans have gathered, chemistry has crossed into cells. LUCA's distant microbial descendants have spread through water, sediment, rock, and air. Photosynthesis has changed the sky. Oxygen has poisoned one world and made another possible. Complex cells have formed through ancient symbiosis. Bodies have emerged in the sea. Animals have learned to move, sense, hunt, hide, and build. Plants and fungi have made land into soil and forest. Extinctions have erased worlds and opened others. Humanity does not stand at the center of this story. Humanity stands at the far edge of it, balanced on a narrow branch made from countless earlier accidents. This is one of the most humbling facts in deep time. The human body is not a clean invention separate from the rest of life. It is an archive. Every breath depends on the oxygen revolution made by microbes more than 2 billion years before any animal existed. Every cell carries mitochondria, descendants of bacteria that became permanent inner companions in the ancestors of eukaryotes. Every gene belongs to a long history of copying, error, repair, and inheritance that began before bones, before shells, before eyes, before even the first confirmed fossils. The calcium in bones, the iron in blood, the carbon in skin, the phosphorus in DNA, the water in every tissue all belong to Earth's older chemistry. A human being is not a break from the ancient world. A human being is the ancient world arranged into a temporary thinking form. The lineage that eventually leads to humans passes through many vanished worlds, none of them designed for humans. Mammal ancestors survive in the shadows of larger reptiles. Early primates move through forested environments shaped by flowering plants, insects, climate shifts, and continental drift. Ape ancestors adapt to changing habitats in Africa. Hominins eventually walk upright, make tools, share food, control fire, develop larger brains, and leave traces in stone, bone, ash, and genetics. But even this human story, often told as though it were the final chapter of evolution, is only the latest visible surface of something much deeper. Homo sapiens is young. Our species is a newcomer on a planet where bacteria have been evolving for billions of years. The microbial world is not our prologue and then gone. It is still here, inside us, around us, beneath us, and beyond us. What survives in the record suggests that human existence depends as much on fragility as on success. Had oxygen never accumulated, large animals like us would not exist. Had eukaryotic symbiosis failed, there would be no cells capable of building animal bodies. Had the Cambrian ecosystems followed a different path, the ancestry of vertebrates might have remained obscure or ended entirely. Had the end Cretaceous asteroid missed Earth, mammals might never have diversified in the same way. Had climates shifted differently in Africa, the hominin path might have taken another direction or disappeared. No single explanation fully accounts for why humans exist, because humans are not the result of one explanation. We are the result of chemistry, geology, climate, symbiosis, extinction, ecology, and chance layered across time. This does not make human life meaningless. It makes it stranger. Meaning does not require being the purpose of the universe. It can arise from being part of an unbroken chain of survival so old that its beginning is nearly invisible. Every person alive is connected, through inheritance, to cells that endured the oxygen crisis, to eukaryotes that carried bacterial partners, to animals that survived ancient seas, to vertebrates that crossed ecological thresholds, to mammals that passed through catastrophe, to primates shaped by forests and climate, and ultimately to microbial ancestors whose forms are lost. The line is not straight, it branches, tangles, breaks, and rejoins through deep ancestry, but it is continuous enough that life today still carries the imprint of beginnings no rock can fully preserve. There is common temptation to turn evolution into a staircase, with humans at the top, but deep time does not support that picture. Evolution has no top. Bacteria are not failed humans. Jellyfish are not unfinished mammals. Ferns are not primitive trees waiting to become forests of another kind. Every living lineage has survived the same span of time since its last common ancestors with others. Some have changed in spectacular visible ways. Some have remained successful by staying small. Some have simplified, some have merged, Some have grown enormous and vanished. Humanity is one evolutionary outcome among many, not the inevitable conclusion of life's story. The more accurate image is not a staircase but a river delta, branching endlessly, with channels forming, drying, merging, and spreading across a changing landscape. Many channels disappear into sand, a few reach the sea, some run underground for a while and return. LUCA is not the spring itself, but a deep surviving junction behind all present channels. Before it, older flows may have existed and vanished. After it, life spreads outward into bacteria, archaea, and eukaryotes, into worlds visible and invisible, into forms that crawl, root, bloom, swim, fly, decay, and think. Humans are one late channel, bright for a moment, carrying water that began far upstream in darkness. This matters for the way the origin of life should be understood. The beginning was not a dramatic moment when the universe decided to make people. It was a threshold crossed by chemistry under conditions still debated, in settings still uncertain, on a planet still dangerous. The first life was probably small, fragile, and hidden. It did not know it had begun anything. It had no future in mind. Yet once reproduction with variation became stable enough, time did what time does. It tested. It erased. It preserved. It allowed rare arrangements to become lineages. Over billions of years, the small became planetary. The invisible changed the atmosphere. The microbial made animals possible. The ancient made the present. Human beings now look backward with instruments no earlier life possessed. We read isotopes in graphite, chemical fingerprints in ancient rocks, genes in living cells, molecules in asteroids, and fossils pressed into stones from vanished seas. We use machines to search for patterns too faint for the eye alone. We compare genomes to infra-ancestors no one will ever see. We study Bennu and ancient zircons, stromatolites and Ediacaran impressions, Asgard archaea and mitochondria, oxygen records and extinction layers. The search is unfinished, and it may always remain partly unfinished. The oldest chapters have been damaged by the planet that preserved enough of them to make us wonder. But perhaps that is fitting. Life began in ambiguity, and our understanding of it still carries that ambiguity. We know enough to see the broad arc, but not enough to remove the mystery. The beginning of life is not a single solved scene, but a field of overlapping clues: water, rock, carbon, energy, membranes, RNA-like molecules, thioesters, minerals, vents, pools, impacts, sunlight, time. Somewhere among such things, chemistry crossed into ancestry. Somewhere, matter began to remember itself well enough that forgetting was no longer complete. And from that first unstable memory, the living world slowly unfolded. The human thread in deep time is therefore both astonishing and modest. Astonishing because a universe of atoms can become aware of oceans, fossils, stars, and its own origins. Modest because that awareness is late, fragile, and dependent on events beyond control. We are not separate from the microbial earth. We are not outside extinction, climate, chemistry, or chance. We are one expression of a 4 billion year continuity that began without witness and may continue in forms no human future can predict. To understand how life began is not only to look backward toward ancient oceans, it is to recognize that every living breath is a continuation of that first difficult persistence, still moving through time. And so the story does not end with humans standing above nature, but with humans listening from within it. The same planet that once held only hot rock and dark water now holds memory, language, science, fear, tenderness, and curiosity. This same chemistry that struggled to organize in ancient environments now asks how organization began. The same life that once survived as hidden cells in a hostile world now turns its attention back across billions of years and finds not a simple answer, but a vast inheritance. We are latecomers, but not strangers. The beginning is still inside us. The long story softens here, not because it is finished, but because deep time has no clean final door. It continues under every field, every sea, every forest, every city light seen from above the night side of Earth. Microbes still divide in hidden sediments. Fungi still thread through soil. Plants still lift sunlight into living tissue. Animals still move through water and air. Human beings still carry within their cells the old partnerships that made complex life possible. Beneath every ordinary day, the ancient story continues quietly. 4 billion years ago, or somewhere near that unimaginable distance, the first successful living systems may have been no more than fragile chemistry, learning persistence in a world of stone, water, and energy. They left no clear face in the rocks. They gave no signal that a future would someday look back for them. Yet their continuity survived where countless other beginnings may have failed. From that survival came oxygen skies, green continents, soft Ediacaran seas, Cambrian eyes, forests, extinctions, mammals, minds, and the ability to wonder. The lesson is not that life was destined. It is that life endured. It endured without certainty, without direction, without protection from catastrophe. It changed the planet and was changed by it. It vanished in many forms and returned in others. It became simple, complex, hidden, enormous, beautiful, strange, and brief. And for a little while on one small world turning around an ordinary star, it became conscious enough to trace its own shadow back toward the first oceans. In the final image, Earth is quiet from far away. No ancient violence is visible. No first cell can be seen. The continents drift beneath clouds. The oceans hold moonlight. Somewhere in dark water, chemistry still moves through living membranes. Somewhere in stone, the old record waits in fragments, and somewhere under the night sky, a living mind breathes oxygen made possible by microbes and wonders how the story began.